Hi everyone, so let's have a quick look at an area of biology where you have to apply your knowledge of enzymes to particular investigations that are carried out. This is quite common for exam questions on enzymes. So let's have a look at what the information is that examiners expect you to know already. You've got two key bits of background information. Something on enzyme structure. So remember the enzyme, often drawn like this. You've got a particular area of the enzyme called the active site, and that's specific. You might have heard of the lock and key mechanism. It's a specific shape, and it's where the substrate binds. That substrate will only bind to that particular enzyme because the active site is a specific shape. The reaction takes place, and you get some products produced. There are also three factors you need to remember which affect enzymes. So temperature, substrate concentration, and pH. There are three kinds of questions you get asked here about the enzyme investigations. Method questions, questions where you've got to do a calculation, and then one where you have to kind of look at the table of results or a graph to interpret what's going on. So let's have a look at the first type. So devise a method to find the optimum temperature for the enzyme lactase. So often with these questions, the first one that they're going to kind of expect you to get is dead easy. Mix equal amounts or volumes of the enzyme and substrate. In this case, the enzyme's lactase, so we should really write that. And lactase breaks down lactose, so we should really write down that as well. Number two, then it says find the optimum temperature. You have to incubate at different temperatures. That makes sense, doesn't it? And then finally, we've got to have a way of knowing the reaction's kind of completed. So obviously, you've got lactose, and it's broken down into glucose. So test for glucose. And you might remember that's using Benedict's solution. Test for glucose, or time how long it takes to produce glucose. Okay, so that's kind of the method kind of questions. Here's a similar one they've got here. This one, instead of temperature, it's about pH. So again, number one, you'd mix the starch solution, because amylase breaks down starch. Mix the starch solution with the amylase, just like before. And instead of temperature this time, you use different pH values. And then test for glucose, or as you know, there's a test for starch, which is the substrate. Test for starch at regular intervals. So they're the kind of method questions you get. The second type of question would be this one, where you've got to calculate rate. So students investigated the time taken for amylase to break down a 10% starch solution into glucose at 37 degrees C. The student repeated the investigation 5 times. Calculate the rate of the amylase enzyme activity for the 10% solution. So they've got lots of numbers there already, but the real ones you want to focus on are the numbers in the table. So if you work out the mean of those, that gives you a total of 588 over 5, which is 117.6. Now, whenever you work out the rate, it's a good idea to do 1 over the time. And in this case, it was 1 over 117.6. So that gives us an answer of 0.0085. Okay, so that's one example of a calculation. Another example of a calculation looks like this. Students investigated the effect of enzyme concentration on the mass of product formed in 1 hour. Now, this is a really easy question. It says, calculate the ratio of enzyme concentration to product formed. They've already told you that the enzyme concentration of 40 units gives you a ratio of 1 to 1. So all you have to do is go to 5 and 15. So the ratio is 5 to 15, and obviously we simplify that, and that will give us a ratio of 1 to 3. So they're kind of straightforward questions, aren't they? Number 5. This one's the final type when you analyze results. Now I just want you to look at two key command words here: describe and explain. So describe is all about what do you see, explain is using the science. So for here, describe the effect, you're actually looking at the graph, and you'd write, as the pH increases, the volume increases and then decreases after pH. Where is it going to start decreasing? It's about pH 7, isn't it? And number 2 for that, we can actually say a little bit more, and that will get the second mark. pH 7 is the optimum pH. So explain slightly different. That's using the science. And in that case, you're thinking about denaturation, aren't you? So step 1, above pH 7, there is a decrease in oxygen produced as the catalase is denatured. So what does that mean? The enzyme's active site changes shape, so you've got less enzyme substrate complexes. There we go. So do keep an eye out for those two keywords, describe and explain.